Hello, I'm Glenn Murphy with Burroughs Wellcome Fund Science Explorers. Today we're here in Raleigh, North Carolina to meet Keith Weninger, Professor of Biophysics at NC State University. Dr. Weninger specializes in single molecule biophysics. That is, he uses custom-built microscopes and lasers to watch tiny proteins and DNA molecules in action, something that would have been impossible just 10 years ago. Dr. Weninger, thank you for joining us.
Oh, thanks for having me.
Cheers. So what exactly is single protein biophysics? How does it differ from, say, biology or biochemistry?
Well, in single molecule biophysics, we literally look at one molecule at a time, and that's different from typical biochemistry that goes on in test tubes, because in a test tube, all of those molecules may not be the same, and they may not be doing exactly the same thing at the same time. And so by watching each one individually, you can learn more about what they're doing.
So what brought you here to North Carolina?
To be part of North Carolina State University. It's a good place for me to do my work, and I like the area because of all the big universities and companies here. It's lots of good opportunities for research.
Great. So do you mind taking us inside your labs, giving us a peek around, introduce us to your team?
Absolutely.
Great, let's go. Wow, so this is a pretty serious looking piece of kit. We've got lasers, we've got mirrors all over the place. What exactly are we looking at here?
These proteins are way too small to be seen by an ordinary microscope, and so that's why we use these tricks where instead of actually looking at it, we measure the color of light that they emit and can use that to tell how they're moving on a very, very short length scale.
So what's the most exciting discovery you've made so far with all this gear?
Well, recently we've been able to see how some of these DNA repair proteins are actually changing shape and moving when they find an error in DNA. And so by understanding this sort of mechanical motion of the proteins and how they move, we get an idea of how they go ahead and repair the errors in DNA.
We've seen how Dr. Weninger studies proteins in action, and he's discovered that DNA repair proteins have unique moving parts that bend and kink DNA as they repair it, in ways that we didn't understand fully before. But why should this matter to the rest of us? What's so important about DNA and DNA repair? DNA carries the coded instructions we call genes, which control pretty much every biological process you can think of. Everything from bone and muscle growth to eye color and fat deposition and fighting off infectious disease. But genes can't copy, read, or translate themselves. The genetic code used in DNA is written in a foreign language, one our bodies can't understand or use without help, and that's where proteins come in. DNA reading proteins are the code breakers and interpreters of the human body. One type of protein copies the coded language and creates duplicate manuals for each new cell as it divides. Each manual contains about 3 billion letters of code. That's about the same as 1,000 full-size Bibles. And as you might imagine, mistakes creep in here and there during the copying process. In fact, about 1 letter per million is copied wrongly, which is really not too bad at all, as it's like copying out an entire Bible and only having 3 typos or misspelled words. Without these proofreading and editing proteins, the code would soon become garbled and the processes of life would break down. It's these editor and repair proteins that Dr. Weninger and his team study in their lab. In fact, faulty DNA repair proteins are involved in about 90% of colon cancer cases, and probably in most other types of cancer too. By better understanding what's going on with these proteins, we could perhaps one day design small, single-molecule drugs that help or replace the function of DNA repair proteins. And as it happens, Dr. Weninger is working with another team at nearby UNC, North Carolina, who are helping to do just that. So the NC State team make the observations, pass the results on to the UNC team, and they make the single-molecule proteins. Together, they're a triangle-wide, cancer-battling supergroup. Not bad, science guys. Not bad at all. Join us next time for more Science Explorers, making discoveries in our world and beyond.